I'm a big diet kind of person. I really like food. I'm really big into food. And so if you know anyone that's really big into food, I would be really familiar with this man right here. His name is Ansel Keys. He's a physiologist. And he did most of his research around the 50s, 60s, and 70s. And he's most famous for his 7-country study, where he determined that countries with higher intake of fats, specifically saturated fat, were more associated with having coronary artery disease. But here's the problem. See, he got put on Time magazine, so obviously he was a very influential kind of person, and that led to this right here. And so, you know, the U.S. government and other governments around the world have been preaching low fat, low fat, low fat, stop your heart disease, but heart disease hasn't been going down. Well, here's the issue. This was his graph right here in his study, but this was the actual data. He used 7 countries, when in reality, he actually had all these countries to go from, and there's actually more data over here on other populations such as the Inuit and other native populations that eat a very high fat diet but don't have any heart disease at all. Very surprising. So, at first, the American Heart Association was very wary of his research, and they actually didn't accept it, and they thought it wasn't good enough to be able to take something and make actual recommendations off of it. But what happened? He bulldozed his way into the AHA, and then he co-authored publications saying that we should definitely reduce our fat, especially our saturated fat intake, to deal with heart disease. And so that led to this. This is Senator George McGovern. He was one of the bulldozers in terms of, oh, there's Amy. So that's Amy. She also helped me with my feeding in public. So thank you, Amy. So this is Senator George McGovern. He's probably one of the most influential people in terms of dietary advice in this nation. He was the one that brought about this whole dietary paradigm that we're in right now. And it wasn't exactly his work that led to this, but in the long run, what happened was we got our first USDA food pyramid that told us that fat should be all the way up here, which is very, very low, and then most of our food should be down here, carbohydrates and stuff like that. So I believe it was 6 to 11 servings of carbohydrates. And he was heavily influenced by Ansel Keys. And at the time, there were obviously a lot of detractors because they thought the science wasn't good enough. There's one more person outside. I don't know if you want to let her in. It's not long. Did you? So, yeah, so that led to this dietary paradigm of just low-fat diet, high in carbohydrate, and I mean, obesity has skyrocketed. I'm not going to say that eating higher carbohydrates and less fat causes obesity. There tends to be more evidence saying that a high caloric intake is more of a cause of that, but we'll continue on. So basically what I'm trying to say here is that America is a very fat-phobic nation, as are a lot of other nations. And even till today, it's still the case. I mean, nowadays it's a little more moderated. We'll say, okay, maybe monounsaturated fats are a little bit better. It's better for curtailing your heart disease and things of that nature because the Mediterranean diet and things of that nature, which Ansel Keys is also a very big part of. So, now here's the problem. Recently, specifically these past couple of years, there's been a lot of publications coming out saying that saturated fat isn't linked to heart disease. Here's one. Here's another one that I found. Here's a third publication by a cardiologist, Asim Malhotra. He published this in BMJ, and he was questioning the evidence that said that dietary fat actually caused heart disease. And here's one last one. So as you can see, there's a lot of conflicting evidence saying that saturated fat doesn't cause heart disease. And you know, here in this nation, we're always led to believe, don't eat fat, don't eat fat. So that's what led to my research. Now, I'm not researching coronary heart disease. I'm just trying to say that here we're very lipid phobic, so we don't eat as much fat as we normally do. I mean, personally, I eat a lot, to be quite honest with you. I love mine. I love mine. Okay, so let's take a look at this. As a public health major, before I continue, I'm a public health policy major, and so I'm not a bio major, so in terms of all the bioterminology, I'm not very well versed, but I thought this was such a fantastic thing to research, so I might as well go through with it, right? So as a public health policy major, I've seen this graph practically millions of times. All my classes, we go over this all the time. 66% of our nation is either overweight or obese, and it's a huge problem. You look around, you can practically count them on your fingers. And here, back in 1991, as you can see, the averages were actually pretty low, 10 to 15%, somewhere around this area. And as the years go on and on and on, you get this. It keeps going higher and higher and higher. And what I remember was in one of my public health classes, his name was Professor Bruckner. What he did was he did it year by year. So what he did was he had a slide, and the first slides were obviously very white, and then he did year by year by year, and it got bluer and darker and redder and more orange. So this is kind of just an extrapolation of a couple of years. So as you can see, this is like by decades and whatnot. So you can see the obesity and overweight problem here in this nation is very, very big. And so that leads to my next part. So what I'm focusing on is medium chain triglycerides, which is a type of fat that's actually been substantiated in the literature as increasing your thermic effect. So like I said earlier, there's a lot of evidence actually saying that overconsumption of calories, which we hear all the time, is actually the cause of obesity and being overweight. And so this is very interesting because MCTs have been shown to actually increase your thermic effect, which is increasing your basal metabolic rate, increasing the amount of calories that you burn throughout the day. And with that, you also lose more, and you expend more calories and you lose more weight. And so here are some publications that I found as part of my research, showing that medium chain triglycerides have this effect. And it's interesting because a lot of these studies actually show that it targets abdominal fat, which has been shown as the most dangerous fat to have on your body because it leads to so many other diseases such as metabolic disorder or heart disease, diabetes, and things of that nature. So that leads to this. So these are the even medium chain triglycerides. So we consider them medium chain because they're in, these are fats. So fats are composed of carbon molecules, obviously, with glycerol at the end. And you have, you can tell they're saturated because there are no double bonds between the carbons. So these are the carbons right here, the little gray ones. There are no double bonds between the carbons, so they're completely saturated. And so they're considered medium because it's between a range of about 6 and 10. So any carbons that are less are considered short chain. Any carbons that are more are considered long chain. Now, there's not really a standard definition of what's really medium because I've seen some papers saying that maybe 10 to 12 can be still considered medium. So, but I'm focusing on here between the 6 and the 10 range. So here we have the even ones, caproic, caprylic, and capric acid, which these ones are usually the ones that are extracted as pharmaceutical supplements and things of that nature. These are the odd ones, C7 and C9. So these are the ones that we're focusing on here in our research. Okay, so why zebrafish? So these are the fish that I'm doing research on. Okay, so when I first got into wanting to do this research, I thought, okay, so I'm going to look at MCTs, MCT oil, coconut oil, things of that nature, right? I'm going to go and test this on humans and everything. I had this crazy big scheme, and then I realized, oh wait, it's probably not going to work. You see, if I did it with humans, it'd be a lot more complicated. I'd have to go through the IRB, et cetera, et cetera, et cetera. It'd be a lot more complicated. So that's when I got in contact with Professor German, super awesome guy again. So I got in contact with him. And he does work specifically with zebrafish. And so I talked to him over a summer session, and then we got this idea kind of hashed together. And again, thank you to CHP for kind of pushing me into this whole research thing, because had it not been CHP, I don't know if I would have gotten involved in research in the first place. So these are zebrafish, and to get kind of a size comparison, let's take a look at this next picture. So if you take a look, that's his finger, and that's the size of a zebrafish. So they're very, very small. So we're talking about millimeter size here. So very, very small. But they're very hardy, apparently. So, I mean, I'm glad. I'm very, very glad. So this is my hypothesis. The fish that are fed the diet with the most MCTs will show the greatest adipose tissue metabolism because of increased thermogenesis. I know that sounds kind of complicated, right? So basically what I'm trying to say is the fish that are fed the most MCTs will burn the most fat. That's pretty much what I'm trying to say, because they have the most MCTs, and MCTs have been shown to increase your metabolic rate. So let's go through how I did it. So this is basically my aquarium system, and mind you, it was very, very complicated to set up, and it took a very long time. So I started setting these up, say around spring break is when we finally got all the tanks together. And if anyone here has ever tried to drill into glass, it's very, very difficult. So if you take a look here, these little black parts are the drains, and they drain into this back pipe right here. So as you can see right there, it drains into the back pipe, and the back pipe goes down into a filter. We call it a sump at the bottom. So we had to drill this hole into these 10-gallon tanks, and it was very, very complicated. It was this enormous process of getting the right drill tip, and then having running water as you're drilling, making sure you don't drill too fast so that you don't break the glass, because once you crack glass, it can crack everywhere. And if I had a count, I think I cracked maybe 3 or 4. And these tanks were very expensive, so it was very complicated. And then once you got them drilled, you had to put these little, these drain covers on them. Doing that was even more complicated because you had to be very fragile with the glass. And as I was trying to put them on, some of them actually cracked afterwards. And so the ones that I thought were good, after trying to fix them in with all the attachments in the back, they cracked again. And I was like, oh shoot, please, you know? So luckily we had a couple safety tanks left over to drill some more and then things of that. So we had to build the entire system. So we had to cut all these pipes right here. We had to cut all these pipes in the back. We had to drill all these holes. I had to cut all the covers for this, cut all these little hoses here so that you could the water could go through. So basically, the entire system runs on universal water, pretty much. So everything is universal water. So that's why we connected them all this way. And if I had to quantify how long it took to put this all together, I'd say it took at least a month. It took a very, very long time. And then I had a lot of help, especially from my girlfriend. She was there to help me, and then Amy and whatnot. So I'm very glad that you guys were there to help me out. So it was a very complicated system, and I'm glad it all got together. So these are the oils that I wanted to focus on right here. So this is an extract of MCT. So it's 100% MCT oil. Specifically, MCT oils are usually, most of them are saturated, obviously. And so this one's all saturated fat, but it's actually liquid at room temperature because it's an extract. Red palm oil has about 5% MCT, and coconut oil is about 60 to 65% MCT. So this extract right here comes from here. So they take coconut oil and they extract the C6, C8, C10 from it, and they get this right here. So obviously it'd be great if I could just feed them the lipid, but for the sake of health, you can't do that to these fish. You know, they gotta get their vitamins and whatnot, right? So we feed them other things because if there were a way, it'd be awesome, but no. So, you know, so the way we made it was we got a food mixer, one of those KitchenAid ones, and we mixed all these ingredients together. So here, for protein, casein and soybean. For carbs, all these ingredients right here. And menhaden oil, so this was for the omegas, the essential fatty acids that the fish need to eat. And so the specific oil right here, this was the one that we made different in every single batch. So I did 7 grams of whether it was the MCT oil, the palm oil, or the coconut oil, and then with everything else the same. So we kept it isocaloric to make sure. So this is how we collected our data. So as you can see, this is spectrophotometry. And the idea is, that word, it's long. So the idea is looking to see how light is absorbed in these samples. And so you can see the increasing lipid right here. So this is just a standard curve that we use to compare. And this is the data that we got right here. So what's very interesting is that I expected the MCTs to be the lowest as a percentage of body weight, but it wasn't. Corn obviously was the biggest, which is very good for my data. Palm was actually the lowest, even though it had the lowest amount of MCT. So this is very interesting. I have yet to figure out why this is. Obviously, this is an N=4, so our P is actually not as low as we would want it to be, so it's not very statistically significant. But there is a trend showing that corn is higher. So the fats with more MCTs are lower, and obviously it'd be better if we had more of a sample size to be able to do it. But we've only been feeding for about 2 weeks now because of all the setting up of all the experiment and whatnot. It took such a long time that we only had 2 weeks to actually feed our fish. So this is what we've got. But so far, it's showing a trend, obviously, with the ones in MCT being lower than the corn oil, which doesn't have MCT. So again, N=4. If we had a bigger sample, it'd be a lot better. So some of the limitations as of yet, like I said earlier, it's a very small sample size, so we really can't extrapolate much out of this. They are isocaloric diets, but the problem is you can't control for how much the fish actually eat. You can control how much you put into the tanks, which is nice, but you can't control for how much they eat, and that's something else that a bigger sample size would be able to help. And again, it's only been 2 weeks of feeding. We plan to do this for about another 6 weeks or so, and then see what data we get later on. But for now, this is what we have. And so Again, I'd like to thank my professor, super awesome guy. I'd like to thank all the all the people who helped me, my girlfriend Amy, and it's everybody else who's been involved with this. And yeah, thank you so much for listening in and questions, right?
Any question? You're finished?
Yes.
Yeah.
Okay. Yeah.
What's the difference? Is there any difference between the palm, other than MCTs, the palm oil?
Yes.
And
Yeah. Definitely.
What is
It depends on the fatty acids. So, for example, if you take a look at say like a coconut oil versus say like an olive oil, right? An olive oil is predominantly monounsaturated fat versus a coconut oil, which is predominantly saturated fat. Obviously, it's not all saturated fat. There's some poly, there's some mono in there, but it's mostly. And so the difference mainly between the coconut oil and say palm oil, there's some saturated fats in palm oil as well. Palm oil, the reason why it's red is because it has beta carotene and also has vitamin E in it. Compared to coconut oil is mainly those medium chain saturated fats, and so that's mainly the difference between different lipids. Some lipids have more saturated fat, some lipids have more monounsaturated fat, polyunsaturated fat. Some lipids have more omega-3s, omega-6, omega-9s, et cetera, et cetera. So that's usually the differences between the fats, and I like to focus my fats more on the saturated side and the monounsaturated side versus the polyunsaturated, which is another kind of quip that I have about, I guess, the American diet is that everyone's using fats that are so prevalent in omega-6s, and they're cooking with them, and omega-6s are very highly oxidizable, and so it's very bad for inflammation inside the body, which might be an even bigger cause of heart disease than we might assume. So, yes.
Because you use zebrafish, how well do you think this generalizes to humans?
Well, at the moment, I don't think it generalizes to humans, first of all, because it's such a short, we only have 2 weeks of study and a very small sample size. I'd rather say that we can maybe extrapolate later on into better animal studies, because zebrafish, it's very preliminary in terms of extrapolating to humans. If you want to extrapolate to humans, I'd say move it on to say like a rodent study or something, which is more appropriate. So there are more rodent studies that are actually more applicable, or other animal studies are more robust that are more applicable to actually human physiology. So zebrafish physiology, it's not as similar to human physiology as other rodents. So you could say that you could generalize to other animals, and then from there, if the data is very significant, you can generalize that over to humans.
How did you measure their lipid?
Oh yeah, this is very complicated. Okay, so I didn't think it would take so long, but it was actually very long. So what happened was, each fish took about an hour, I have to say. So n equals 4, right? So it was a very long time. So what we did was we extracted the fish, humanely killed them, and then what we did was we had to grind them. We ground them in a 9-to-1 solution with a deionized water. And then from there, we took about 10 microliters of that. We mixed that with a chloroform methanol, which is a solvent solution, to kind of extract the fat from that. From that, what we did was we vortexed it, added simple salt water solution. Then we did a centrifuge, and then from there, that's what we can get the fat. We baked it so we can remove all the solvent. And then what we did was we put 36 molar sulfuric acid into it. And then from there, you could char the carbons. And that's when we go back to the, we go back here. Go back to this right here. You char it, and you see how much is charred and how much light actually gets reflected in there. So that's how we measure the lipids. Very complicated process, but yeah.
I have a question.
Yes.
As you mentioned in your presentation that heart disease is because the diet is increased in America and around the world.
Correct.
And also, as we know that there are 2 types of lipid. There are cholesterol and there are triglycerides.
Correct.
And then, do you think which one more dangerous?
Which one's more dangerous?
Yeah, triglyceride or cholesterol?
I would say, well, it really depends. I wouldn't say there's conclusive evidence to show that cholesterol actually causes heart disease. I don't believe it does. We're led to believe HDL is the good, LDL is the bad, but really these are just transporters. All cholesterol is the same. HDL is just a transporter, LDL is just a transporter, things of that nature. And so when people say cholesterol, I'd say it's only, cholesterol is bad in the sense if it gets lodged in your arteries, and usually what happens is if you have a lot of inflammation in your arteries, then that would be a bad case, because then the cholesterol has to come, because cholesterol is an antioxidant. don't realize that. People would be happy to say, oh yeah, I have 0 cholesterol. They'd be super happy with that. But really, without cholesterol, we'd all be dead, to be honest with you. And so cholesterol is an antioxidant. And so with the inflammation in our arteries, that's what happens. The cholesterol gets led there to kind of, they're more like the firemen to kind of quell the fire. But the problem is with all the excess inflammation, what happens is they might get lodged there, and then that might cause coronary blockages. And so that's why a lot of people say that having a high cholesterol might be a problem. But if you have a high cholesterol, you have low inflammation, I say that's a little bit less of an issue, simply because